Good afternoon to all the NEET aspirants. This is your Prasanna Ma'am from NEET World. So yes, we are here to discuss the questions today from photosynthesis in higher plants, and the topics we are going to cover today are C4 cycle and CAM cycle. So yes, before going to the questionnaire session, I would like to brief you or give you a gist on the courses that we are running in the NEET World. So in the NEET World special program, we are running 3 programs, 2 steps to medicine, wherein this is for 11th standard students who are going to write NEET 2027, and 1 step to medicine, that is for 12th standard students, and NEET World's power step to medicine, this is for long term students. The complete fee structure for all these 3 programs is just 3333 for month. Okay, it is just 3333 for month. So you can contact to the given number over here to get further more details about this. And next, so the programs which I have just told you are starting from July 1st. So here in our offline coaching center, we have well equipped classes, and the material is provided both for online and offline students. Both online and offline facility is there. For online students, you can install NEET World app, which is in the Play Store, and the classes will be run as usual for you. Materials will be same for both of them, and for offline students, hostel facility will be available both for boys and girls. Now let's start our questions. So here you look into this, read the following statements and find out the incorrect statement he is asking us. Okay, to find out the incorrect statement, you look into this statement A. Second step of Kelvin cycle, that is reduction, involves utilization of 2 molecules of ATP for reduction and 4 NADPH for phosphorylation of 2 molecules fixed. To fix 2 carbon dioxides, first you have to recall for 1 carbon dioxide during the time of reduction, 2 ATP and 2 NADPH are required, right now beta. But for 2 carbon dioxide it is 4 ATP and 4 NADPH, it is not 2 and 4 NADPH. So this will be the wrong statement. Of course, we have identified the incorrect statement, but let us look into the other 3 statements as well. The regeneration statements required the regeneration steps require 1 ATP for phosphorylation to form RUBP. True? Yes, per 1 carbon dioxide 1 ATP is required to synthesize to regenerate 1 RUBP. It is probably to meet the differences in number of ATP and NADPH used in dark reaction that the cyclic phosphorylation takes place. Yes, this is correct only. The difference in the energy is there. Of course, for 1 carbon dioxide in total in Kelvin cycle, 3 ATP are required and 2 NADPH, right ma? Next, plant that are adapted to dry tropical regions have C4 pathway. All the tropical plants, C4 plants are what? Tropical plants. C3 plants are what? Temperate plants. We have already discussed about it now, ma'am. Here you see this question. Malic acid, that is 4 carbon, is produced in which plant without Kranz anatomy. So see, he is saying 4 carbon malic acid is produced. Yes, 4 carbon malic acid is produced and there is no Kranz anatomy. Then obviously it is CAM cycle, nama. Obviously it is CAM cycle. What is the only difference between C4 cycle and CAM cycle? In C4 cycle, both mesophyll cells and bundle sheath cells are involved in carboxylation. Fine. Whereas in the CAM cells, only mesophyll cells are involved in the carboxylation process. And second is in C4 plants, Kranz anatomy will be there for bundle sheath cells. CAM plants do not exhibit any Kranz anatomy. And most importantly, in CAM plants, the carbon dioxide fixation takes place only during night time. Okay, carbon dioxide fixation takes place during night time. So carbon dioxide fixation during night time, this is because of what? scotoactive stomata. This is because of scotoactive stomata. What does the term mean? Scotoactive stomata. Scotoactive stomata are nothing but the stomata which opens during night and closes during daytime. Clear ma? So, CAM plants will fix the carbon dioxide during night time, wherein this is because of what? Scotoactive stomata. Clear na? These are the primary differences which I have told you. So, for Cran's anatomy examples, best examples, Opentia is well noted example, we all know that. Along with that, we have to remember Bryophyllum plant and Kalonchi plants as well. Okay, Bryophyllum, Kalonchi, Opentia plants, these are the examples for CAM plants. Okay, this will run what cycle? CAM cycle. To fix 4-carbon malic acid. Clear nama? This you see, as compared to C3 plants, how many additional molecules of ATP are needed for net production of 1 molecule of hexose? Net production of 1 molecule of hexose means what ma? 1 molecule of glucose he is talking about. 1 molecule of glucose by C4 plants. How much additional ATP is required he is asking. In Kelvin cycle, we all know for 1 carbon dioxide 3 ATP are required, but in C4 cycle additionally for 1 carbon dioxide fixation 2 ATP are required. Okay, 2 ATP are required additionally along with the 3 ATP which runs the Kelvin cycle. So, for 1 carbon dioxide it is 2 ATP, then for 6 carbon dioxides are involved for formation of glucose, right, we know. So, for 6 carbon dioxides how much ATP will be required? Okay. So, it is just 6 into 2, that is 12 ATP. 12 additional ATP are utilized by C4 plants to fix the carbon dioxide. So, it is what? Option C. C will be the right answer. This you look into ma, again a statement based question. Photosynthesis in C4 plants is relatively less limited. Okay, photosynthesis in C4 plants, it is less limited by atmospheric carbon dioxide. So, photosynthesis in C4 plants is less limited by atmospheric carbon dioxide because of what reason he is asking. First you remember the primary carbon dioxide acceptor in C4 plants. What is the primary carbon dioxide acceptor? The primary carbon dioxide acceptor in C4 plants is what ma? PEP, and this is catalyzed by the enzyme peptidase. Okay, peptidase will accept only carbon dioxide. So, you remember this. Of course, we have already discussed this in the factors affecting photosynthesis, but once again I am telling you, in C4 plants the cycle runs more efficiently at low carbon dioxide concentration, where it is maximum 360 microliters per liter, and at high temperatures, okay, and at high light intensities. Take it down ma, this is very important. Fine. C4 cycle runs more efficiently in low carbon dioxide concentrations, approximately 360 microliters per liter, and high temperatures and high light intensities. Clear na? And the primary carbon dioxide acceptor PEP, and it is catalyzed by peptidase which accepts or it will react only with carbon dioxide, unlike Rubisco. Okay. So, here you look into the options carefully. Effective pumping of carbon dioxide into the bundle sheath cells. This will not be the reason for photosynthesis of C4 plants with respect to less carbon dioxide. This is not the reason. Rubisco in C4 plants has higher affinity for carbon dioxide because Rubisco is where ma? In C4 plants it is in bundle sheath cells. This will fix the second carboxylation. This will conduct second carboxylation in C4 plants and bundle sheath cells. So, here we are talking about primary carboxylation. So, this statement will not work out. 4 carbon acids are primary initial carbon dioxide fixation products. Yes, this is true statement only, but this is not applicable to the question. Last option you see, primary fixation of carbon dioxide is mediated by a PEP carboxylase. This is the correct answer because pepcase will accept only carbon dioxide, it will react only with carbon dioxide, not with oxygen, unlike Rubisco. Okay. So, as it is only reacting with carbon dioxide concentration should be low. Clear ma? Next. Bundle sheath chloroplast. Bundle sheath chloroplast of C4 plants will lack what? We all know bundle sheath chloroplasts are called as agranule chloroplast. These are called as what, Nana? Agranule chloroplast. This agranule chloroplast will not have grana. So, on grana what photosystems you will be having? PS1 and PS2. Here grana is not there, so it does not have PS1 and PS2. Okay. But then it has stroma and stroma thylakoids. It has stroma and stroma lamellae. Okay. Stroma lamellae is present. We all know over stroma lamellae, what is present? PS1 is present. PS1 is present in stroma lamellae. So, here what will be the right answer, ma? PS2 is not present because those are agranule chloroplasts, grana is not present. So, there is no PS2. There is no PS2 means there is no photolysis of water also. Photolysis of water also in the bundle sheath cells. PS1 will be present, PCR cycle, nothing but C3 cycle. It will be conducted and photophosphorylation will happen. But then what is lacking in the bundle sheath chloroplast? That is photosystem 2, and photolysis of water will not take place. Clear, Nama? Next question you look into assertion and reason type of question. Plants utilizing first RUBP in carbon dioxide fixations are called as C3 plants. True. Okay. Plant will utilize first RUBP in carbon dioxide fixation are called as C3 plants. True. Next, plants utilizing first PEP in carbon dioxide fixations are called as C4 plants. Both the statements are correct, but here no way reason is explaining the assertion. So, in this case what option we have to opt? A and R are true, but R is not the correct explanation of A. Next question. This you see, CAM plants do not show photorespiration due to what reason? Keeping stomata closed during daytime. CAM plants do not show photorespiration due to keeping stomata closed during daytime. This is correct only. Using PEP carboxylase, fixing carbon dioxide into organic acids in night and releasing carbon dioxide in day, performing Kelvin cycle at night. This is wrong. Okay. It will not perform Kelvin cycle at night. It will perform Kelvin cycle during daytime. So, here CAM plants do not show photorespiration, that is because fixing carbon dioxide into organic acid, that is oxaloacetic acid in the night time because of scotoactive stomata. Scotoactive stomata and releasing carbon dioxide during daytime. This will be correct. Here option A is a true statement, but this is not completely explaining the question. Keeping stomata closed during daytime, this is only talking about stomatal opening and closing, and here it is talking only about utilization of PEP carboxylase. This is the statement option 3, wherein it is completely explaining fixing carbon dioxide into organic acids in night and releasing carbon dioxide during daytime. So, option C will be the correct answer, ma. Next, CAM occurs in thin green leaves, wrong. Thin green leaves with parallel venation, wrong. Thin colored leaves, wrong, because CAM cycle occurs mostly in the desert plants where leaves are thick and fleshy. Okay. CAM cycle will occur in most of the desert plants where leaves are thick and fleshy. Done, ma. So here what option goes true? Option D, fleshy green leaves. Clear? Next question. Assertion reason type of question you see. Cam plants will lack structural compartmentation of leaf as found in C4 plants. Okay. Cam plants will lack what? Structural compartmentation of leaf as found in C4 plants. True. Stomata of cam plants are open during daytime. This is wrong. Stomata of cam plants will be open during night time. And here there is no structural compartmentation as that you see in C4 plants, like how mesophyll cells and bundle sheath cells. Here everything occurs in the mesophyll cells only in cam plants. Everything occurs in mesophyll cells. Clear? So here assertion is true and reason is false. Option C will be the correct answer. Next. Select the correct statement regarding first stable product formed in Hutchins-Slack pathway. Hutchins-Slack pathway, you will also call it as what pathway, ma? C4 pathway. Okay. So here OAA, that is oxaloacetic acid, is formed by carboxylation of PEP in bundle sheath cells. Wrong, because the first carboxylation will occur in where, ma? Mesophyll cells. So this is wrong. OA is formed by carboxylation of PEP in mesophyll cells. This is true. Phosphoglyceric acid is formed in mesophyll cells. Wrong. OA is formed in mesophyll cells. Phosphoglyceric acid is formed in bundle sheath cells. Yes, it is formed during what? Kelvin cycle. But here the correct statement he asked about the first product. First product is oxaloacetic acid. So here we have to go with what option? Option B. Sometimes in statements, ma, two statements will be correct only. Okay, like how we have seen here, D is correct, B is correct. But which statement is relevant to the question, that you have to choose. Okay. Next, this you see beta. Which of the following cells will lack Rubisco enzyme in C4 plants? Which cells will lack Rubisco enzyme in C4 plants? Bundle sheath cells will be having Rubisco, because of which it is conducting C3 cycle. Mesophyll cells will lack Rubisco, but then they have what? Pepcase in C4 plants. So the right option will be B. These are simple questions only. Cran's anatomy is not exhibited by which of the following plants? By which of the following plants Cran's anatomy is not exhibited? Cran's anatomy is seen in what? C4 plants. The best mnemonic which I can give you is SMS to remember few examples of C4 plants, that is sugarcane, maize, or most of the millets you can say, and sorghum. They conduct what? C4 cycle. So this C4 cycle or Cran's anatomy, it is not exhibited by which of the following plants? This is not exhibited by sunflower, because maize, sorghum, sugarcane, in them C4 cycle is activated. These are what plants? Tropical plants. That's it. So here what will be the option? Option D. Next, in C4 plants, Kelvin cycle operates in. Where does the Kelvin cycle operate in? C4 plants. Okay. Kelvin cycle operates in bundle sheath cells. Bundle sheath cells will not have grana, right? So agranal cells of bundle sheath cells. That is stroma of bundle sheath chloroplast. Option A. Here if you see grana, it will not occur. Grana of mesophyll chloroplast, it will not occur. Stroma of the mesophyll chloroplast also, it will not occur. It occurs in stroma of the bundle sheath chloroplast. Yes, this question you see, ma. Yeah. So choose the correct combination of labeling 1 to 5. 1 is what labeling? This cell is what cell beta? Mesophyll cell. And 2, if you see, what is this cell? Bundle sheath cell, right? Fine, ma. And here you see the primary carbon dioxide is fixed as is converted to hydrogen carbonate by the enzyme carbonic anhydrase, and this hydrogen carbonate along with PEP, which is the primary acceptor, will form what? Oxaloacetic acid. This is step is called as what? Fixation. So 3 will be what, Nana? Fixation. And then C4 acid, which is over here, this got decarboxylated to C3 acid and carbon dioxide. So 5 will be what? Decarboxylation. Same case you see over here, C3 acid is transported back. Okay, this is what? Regeneration. Now you choose the correct answer. Like 1 will be mesophyll cell, 2 will be bundle sheath cells, 3 is fixation, 4 will be regeneration here. 4 is regeneration and 5 will be decarboxylation. So what will be the right option beta? This one, wherein this is a mesophyll cell, 2 is bundle sheath cell, 3 is carbon dioxide fixation, 4 will be regeneration, 5 is what? Decarboxylation. Clear, right, ma? Next. Carbon dioxide, NADPH and ATP. This ratio he is asking in C4 plants. In C4 plants, let us suppose for 1 carbon dioxide, of course, how much NADPH is required, ma? Same, 2 NADPH as you see in the C3 cycle, okay? And ATP it is 5 ATP on total, right? 5 ATP are required. So when you have to take the ratio for carbon dioxide, NADPH and ATP, it will be 1:2:5. It is as simple as it is. If you want to go for 1 glucose molecule also, for 1 glucose molecule, it is 6 carbon dioxide, 12 NADPH and 30 ATP. If you take the ratio of 6:12:30, then it will be again 1:2:5 ratio only. Clear, ma? This question you see, which one of the following crop plants is very efficient converter of solar energy and whose net productivity value ranges from 2 kgs to 4 kgs per meter square or even higher? Don't get confused with the question. The entire motive of this question is in which plants efficient photosynthesis will occur, or in which plants efficiently glucose is being synthesized, where net productivity is higher. That is the motive of this question. We all know that the net productivity value is higher for C4 plants. It is higher for C4 plants when compared with C3 plants, right, ma? When compared with C3 plants, in C4 plants the net productivity or efficiency is higher. So in this case, what options you have to choose, beta? Yeah, which one? That is sugarcane and maize. Yes. Wheat is again C3, yes. So here the option will be A, C4 plants. Okay, these plants net productivity or the conversion of solar energy completely without photorespiration will happen. Clear, beta? Next. Yeah, this question you see, last question if I am not wrong. Yeah, this question you see, beta. Consider following statements with respect to C4 pathway and select the correct ones. We have to choose the correct statement, okay? Mesophyll cells will possess both rubisco and pepcase. Mesophyll cells will possess, yes, both rubisco and pepcase. Wrong. C4 plants, right? mesophyll cells will possess only pepcase, not rubisco. Initial carbon dioxide fixation occurs in the mesophyll cells, true. Final carbon dioxide fixation, that is second carbon dioxide fixation, will happen or will occur in the bundle sheath cells. So, here statement 2 and 3 are correct. Here he asked us for the correct statement, so 2 and 3 are correct. This we have already done, ma. This is a repeated question. Yes. I would like to end the session by saying you the same points about NEET special program. Make use of it because it is with very much minimal fee, affordable for all sort of students from all financial backgrounds. For 11th students of 2 steps to medicine, 12th students for NEET World's 1 step to medicine, and long term students for NEET World's power step to medicine. All these 3 programs are with the same fee, that is 3333 per month, okay, with complete study materials. For more details, just call on to this number or directly visit our branch. This is the address. So, for offline students, hostel facilities are available both for boys and girls, and this is an AC campus with digital and AC classrooms we have. And the most unique feature about NEET World is one-on-one mentorship for students. Each student is carefully nourished over here. So, again about the fee structure, and these are the features that we are providing from the NEET World. Most importantly, I would like to say that new batch is going to start from July 1st, 2026. Whoever are interested, enroll for the batch by calling on to the given number. And one more thing to say is NEET World's Telegram group. In this Telegram group, daily quizzes are given for biology, chemistry, physics and maths. Live class schedules will be updated in the YouTube live classes. Free resources of materials are provided. One-on-one doubt solving session with the faculties are also given if you call on to the number given. Clear? So, the NEET World will be your ultimate destination for NEET, JEE, MCET and CUET entrances. Clear? Thank you. See you in the next class.